So our next speaker is Dr. Brandon Gallas from the Food and Drug Administration, who's going to speak to us on the regulatory mechanisms and tools for software as a medical device, a subject close to my heart. But I'd like to just remind everybody, please, if you could, to post your questions in the Q&A section of the chat, so that we have something to discuss in the Q&A session at the end. Thank you. Over to you, Brandon.
All right. I think you handed me the option to share my screen. Is it being shared right now?
Yes, I think so, yeah.
You can see that's full screen?
I can, yeah.
Great. So I'm very happy to be here today. Thank you for inviting me. I'm glad to hear some of these talks that have already happened and looking forward to the rest. My name is Brandon Gallas. I'm from the Food and Drug Administrations in the US. I'm in the Division of Imaging, Diagnostics, and Software Reliability. And I'm going to give you a little tour of some regulatory mechanisms and tools for software as a medical device. And of course, we're focused on AI and imaging, and specifically digital pathology. So these are the four main topics, and they're pretty distinct. So don't think that one depends on the other. I'm going to give you a little overview of how we look at software as a medical device and some history. I'm going to talk about a new framework.
Can you make it full screen, please? Make it full screen.
I asked if it was, and I'm on full screen on another screen, so I wasn't able. Let me try resharing again. Can you do that again, please?
Sorry, Brandon, my mistake.
Yes, I forgot to pull down to pick the screen. Right, I am going to do this screen. Is it full screen now?
Excellent.
Yeah. Yeah.
Great. Great. Like I was saying, we'll also talk about change control plans, which is a new framework at the FDA for algorithm submissions that actually aren't submissions. They're part of the pre-baked plans of an original algorithm that will be updated. I'm going to talk about our medical device development tool program and my high throughput truthing project. So these are four distinct situations. And so obviously we're all here because of algorithms and digital pathology, and they're really transforming healthcare in many different areas. And the key is the data, and not just the data, but the curated data, the annotated data that we saw on David Sneed's talk, and real world data that needs to be cleaned up, and with all our natural language processing tools and so on. And really, when we think of software as a medical device, it's not just in imaging, it is across all technologies. We're seeing it in every division almost at the FDA. So I want to give you that a little bit of history, because radiology has been doing algorithms for almost 2 decades now, and we have a little bit of a lingo in the space of radiology. Whether that will translate into pathology is not important, but I wanted to at least give you a heads up to some of these terms in case you encounter them in other areas. We think about CAD as computer aided detection. If it has an E at the end of it, and CADX is when we're talking about diagnosing something selected by the clinician. And then obviously an algorithm could detect something and then provide a diagnosis on top of that. Some of the newer ways algorithms are being used is to triage cases. So the algorithm processes the images and moves images or cases that it feels are more dangerous and needing more attention into the top of the list in the clinician's workflow. But there's also quantitative imaging is a large area where we're just trying to measure different features in images. And then a really interesting area is using artificial intelligence for image reconstruction and noise reduction. I never would have thought that that would have been a use case, because I'm so used to you know, the math and the physics related to the image formation process. But now they're throwing a lot of that away in some spaces to do image reconstruction by deep learning. It's pretty amazing. And obviously segmentation is a big tool in artificial intelligence. I wanted to point to a wiki page that I have developed over the years in responding to questions from people. Kind of really, it's just a list of guidance documents and other FDA materials and some examples of decision summaries with the special controls. Special controls are basically the information we want to see in submission. So that's a nice place to get started if you have no experience in submitting anything to the FDA. And there's also some links to presentations in the space, some of them regulatory, some of them more researchy. So my regulatory advice is always talk to the FDA early. We have a program that allows us to hear about pre-submissions. And so please search online for pre-submissions meetings at the FDA. That's the best way to tell the FDA your plans. I also usually recommend manufacturers start with a very narrow scope. If you try and clear a device for all input acquisition systems and all the features that are interesting and a lot of fun graphics, all of that needs to be validated. And if you try and do that in the very first submission that's very new to us, it's going to be a challenge. So I often recommend that the first submissions kind of limit to the key clinically most useful information you can get to the clinician. And then if you do that, you can expand the indications over time to other imaging systems and protocols, other updates and improvements, adding new features and graphics or metrics. And this is probably less burdensome, possibly less burdensome, because the FDA already knows about the device and has a baseline for the performance. And what I mean by less burdensome, I'm talking about using technical arguments or phantoms and simulation, rather than a patient base. And you can maybe reuse cases from your original submission, but now you've got the new features, and maybe that will still require a new study, a reader study, where the clinicians have to interpret those features. Or maybe the studies can be much smaller with fewer readers. The more information we have in our collective history on a device, the easier the evaluation is. And this means standalone performance. I use the idea of readers. That's when the reader is in the loop when we evaluate a system, and the clinical task then has to be performed by the reader. And that's what we call a clinical study. And then the standalone performance study is really just a head-to-head comparison of some reference standard to the algorithm. And perhaps in these less burdensome methods, you won't need to do much statistics. But it all depends on the risk of the device and what we know about the device already. So I'm going to switch gears a little bit now and talk about change control plans. In 2019, we published a white paper on a framework for change control plans, or for how to update algorithms and control them to keep them safe and effective. And the idea that we're trying to develop is to have a framework for algorithm updates that don't actually require a new submission. So you can imagine how interested the industry is in this, to stop having to submit and wait for reviews and stress over the decisions. But this is not a required element of any algorithm submission. It's just an opportunity to perhaps lay the groundwork for future changes. And the two big pieces in this framework is the SPS, or the software or SAMD pre-specifications. This is really a detailed description of what will be changed. And then partnered with the SPS is the ACP, or the Algorithm Change Protocol. And this is a description of how the algorithm will change while remaining safe and effective. And I'll get into both of these pieces in a moment. So here we are with the SPS. Please be specific. This is a list of algorithm changes that you plan to make. It's not opening the door to do all sorts of research and development and try and optimize your algorithm without ever seeing the FDA. It's a chance to say you have a plan for making changes, and here's how you will implement those changes. And the real prototype examples of such changes is retraining on more data. You know, before you're on the market, you don't have access to as much data, but when you're on the market, you do have access to more sites and also the wide spectrum of cases. And so that's one of the reasons to retrain, is to allow for building a training set with more diversity. This includes patient subgroups or expanding indications to new acquisition devices. And the reason is that you might not have that opportunity before your device is on the market. And that's really the spirit of the changes we're looking for in this framework. So that means that changes like the following here are not really specific enough for us to know what it will look like over time. You know, a change that is described as an update to the algorithms to improve performance doesn't tell us anything about, is it an update to the loss function? Is it an update to the layers, the architecture of the algorithm? Is it an update to the outputs? What exactly is changing is what we need to know, so that there's some kind of limit to the scope of the changes. And we don't want to be dealing with unended, unbounded changes. So moving on to the algorithm change protocol, like I said, it's partnered with the SPS, and each item, each change you're planning in an SPS should link to an algorithm change protocol. And this is really, this is basically the core of what's in any submission anyhow, and we're just asking that it be kind of given up front so that we know what's going to happen. Everything about your data management practices, how you're going to change the algorithm, what are going to be your criteria, your gatekeeping for making sure the performance of the algorithm is adequate, and then how are you going to release this and how are you going to communicate the release to the users is very key. Like I said, it's really everything we would see in a submission, only it's maybe ahead of time before the change happens. And this is my short list of items that I keep over and over again having to tell industry to add. You know, we're not just looking for your framework for these changes. We want to know the specific items. You know, how many sites are you going to be involved in this changing? How are you going to do your truthing, your labeling of your reference standard? And tell us the size of these data sets that will motivate a change, either the size of your training set or the size of your testing set. Numbers is what we're looking for here. The actual statistical endpoints, you know, list us the primary and the secondary endpoints that you will use in your gatekeeping for performance evaluation before releasing an algorithm. I particularly like when we see how the algorithm is changing. So that's comparisons to previous data sets and algorithms, but also kind of an absolute performance on a new test data, so that we really know that at a level we're not going to always base everything on a comparison. We're going to look at absolute performance metrics as well. So please give us the numbers. Please account for uncertainty. You know, error bars are what the FDA are all about, and identifying correlations. When you have a patient, you have multiple images, or you have multiple regions of interest in an image, those are all correlated, and those correlations are often overlooked in the literature that I've seen. And then compare and contrast how your device is with the previous original device. All right, I'm moving on to a different program now, very distinct from what I've talked about so far. This is just like the framework I just talked about, which is totally optional. This program is a voluntary program, but we're hoping that it really helps us with the evaluation of devices. A tool, instead of a medical device, we're talking about development tools, or really it could have been also called evaluation tools. They're a method or material or measurement used to assess the effectiveness, safety, or performance of a medical device. And it really hinges on the context of use. The context of use tells us how that tool should be used. It talks about the output and measurements from the tool, and it really couches it in the device evaluation process. Where does it live in a regulatory submission? That's what the context of use gives us. So that's everything for the FDA. It helps us narrow our questions and needs. The qualification process really means that the MDDT has been validated and it can be used without discussion. The FDA should be comfortable using it. Here's a slide. It's two-phased. There's a proposal about how you're going to collect the evidence, and then there's the qualification package. This slide has a bunch of links for you to refer to later. I'm moving on now to my research project, which is referred to as the High Throughput Truthing Project. A lot of elements of what I saw from David Sneed's talk about annotation is key. And this is our entry portal for our collaboration information you can look at later. But the goal really is to collect pathologist annotations to serve as the reference standard and to pursue one of these MDDT qualifications for the slides, the images, and the annotations so that they can be used by industry to validate an algorithm and put the results in a submission. And the nice thing is the manufacturer won't need to design the study, won't need to collect the data. They only need to compare their algorithm to the annotations in a prescribed way. So that should speed things up, at least in this specific application. Obviously, data is very application specific. And on the right-hand side, I'm showing different images of tumor infiltrating lymphocytes in breast cancer. That's the biomarker that we're annotating right now, creating a reference standard. Many of you might know that TILs are prognostic for survival. And we really believe once the TILs evaluation gets into the patient record regularly, it will be used to inform patient management. And really the goal is to reduce the use of toxic chemotherapies, because the more TILs we see, the better the prognosis with the patient without any therapies. So that's something we're looking forward to. And we imagine a software as a medical device to do this task, to reduce the burden on the pathologist and to help the patient get measurements that are reproducible and quantitative, and so that we can really establish thresholds for making patient management decisions. I don't want to talk too much about the pilot study, except to say that we have a significant pilot study data set right now. These are the specifics. You can look at them at a later date, but you got images, you have ROIs within images, and pathologists are scoring them. To get more information, we have put all the pathologist annotation data online. It's at GitHub. It's an R data package, so statisticians should be able to download it and work off of it. And I'm hoping to inspire statisticians to do that so that we can get more diverse opinions on the evaluation methods and estimating pathologist agreement and then algorithm to pathologist agreement as our performance mechanism to be appearing in submissions. So where were we in the project? Our next steps are to really update pathologist training. We have a lot of pathologist variability in measuring this biomarker, so we see the need to really build up our pathologist training. And we're going to continue with the pilot study just to stress test the, we have a microscope system to collect the data, and it got kind of sidelined during the pandemic, so we're still making sure that that's all up to snuff. But we also have 2 digital mode data collection portals that we have been collecting data for the pilot study. We're finalizing it on the pivotal study statistical analysis plan so that we can size it properly and know our statistical methods ahead of time. We're getting feedback from the community, including the FDA reviewers of this MDDT submission. And we have our research collaboration agreements executed, so we have just started to source and curate our pivotal study slides. That, as you all know, the agreements and the sourcing is a big undertaking. But we're doing it. Most of this is a voluntary effort, and I can't believe and the collaborators that are involved. Very happy that they are helping. And then we will plan and execute our final data collection. So I shared some specific programs and research that are happening at the FDA. I hope these main items came across, and this is just a summary of them here. You know, a lot of these extra elements are voluntary or the research project and not required in all submissions. So please don't be confused that everything I said today is expected. I want to give a last shout out to those collaborators I mentioned, because I wouldn't be anywhere without their help. And I also want to point out some of the more organizational elements that have been helping me along the way. That last piece was the FDA involvement in a collaborative community. So that's an official relationship that allows interface between external collaborators and industry and academia and the FDA. And I appreciate the chance to say all that today. Thanks.
Thank you.